Welcome back, everyone, to another deep dive into the latest and greatest in longevity research. We've got 3 preprints fresh out of the oven, and trust me, you don't want to miss these.
Definitely some exciting stuff coming out right now.
For those of you just tuning in for the first time, this is a deep dive tailored specifically for longevity biotech researchers and experts. So we're going to skip the basic explanations and jump right into the meat of these papers.
Exactly. We're talking publication dates, authors, journals, institutions, and most importantly, why these papers are making waves in the field.
Right. We'll analyze the heck out of them, connect the dots with other research, and hopefully give you some food for thought for your own work.
Sounds good to me. So what's first on the menu?
All right, let's kick things off with a paper from bioRxiv. It's titled FM162A, a mitochondrial gatekeeper of bioenergetics, driving cellular protection and longevity.
Okay, mitochondria, always a hot topic in aging. What's the scoop on this one?
Well, it just hit bioRxiv a few days ago, authored by Madame Moros and a team from Universidad Andrés Bello.
Interesting. And what's this FAM162A all about?
It's a mitochondrial protein. And get this, overexpressing it in Drosophila, those food flies, led to some pretty significant results.
Oh, Drosophila. Always fascinating to see what they uncover with those little guys. So what kind of results are we talking about here?
Longer lifespans, like way longer, and better resistance to heat stress, too.
Sounds like we're talking about potentially hitting 2 birds with 1 stone lifespan and health span. That's impressive.
Exactly. It's a 30% increase in median lifespan.
Yeah.
Not something to ignore, right?
Not at all. A 30% increase is pretty substantial, especially in a model organism like Drosophila. Makes you wonder about the potential implications for humans.
Right. It does make you think. But, flies aren't exactly tiny humans, are they?
True enough. There's always that leap of faith when extrapolating from model organisms to humans. Still, a 30% increase in lifespan in flies is a pretty strong signal that something interesting is going on with this FM162A protein.
Totally. And it's not just about living longer. It seems like FM162A also plays a crucial role in maintaining those mitochondria, impacts their structure, how they produce energy, even cell survival.
No, that's the kind of stuff that gets me excited. If we're talking about preserving mitochondrial function, we're talking about potentially delaying a whole host of age-related issues.
Absolutely. And they even dove into the molecular mechanisms, which is, always the good stuff.
All right, spill the beans. What did they find?
Well, it seems FM162A is involved in this intricate dance with another mitochondrial protein called OPA1, the one involved in mitochondrial fusion.
Yeah, OPA1. That's a key player in mitochondrial dynamics. So how does FM162A fit into all of this?
They haven't completely nailed down the exact relationship, but it seems like FM162A might be regulating OPA1 activity. And you know how crucial that balance between fusion and fission is for those mitochondria?
Right, that delicate dance between merging and dividing. It's essential for keeping those mitochondria healthy and functional. When things get out of whack, well, that's when we start seeing those age-related declines and diseases popping up.
Exactly. So if we can figure out how to modulate FM162A, could we potentially be looking at a way to keep those mitochondria humming along smoothly for longer?
It's definitely a promising avenue for research, lots of potential there. But let's not get ahead of ourselves. This is still early stage research. We need to see how this all plays out in mammalian models before we start popping the champagne.
Fair enough. But it's exciting to see this kind of research coming out.
Absolutely. And speaking of exciting research, what's the next paper on our list? I keep thinking about that 30% lifespan increase in the flies from that first paper. That's a pretty significant jump, especially when you consider it's just from manipulating a single protein.
Yeah, and not just living longer, they seem to be healthier longer too, right? The flies with increased FM162A maintained better locomotor function as they aged.
Right, suggesting that it's not just about extending lifespan, but also health span. And that connection to the mitochondria, their structure, bioenergetics, it's all very intriguing. We already know how crucial mitochondrial health is to the aging process. Could this FM162A be a key piece of that puzzle?
Well, the paper did dive into some of those molecular mechanisms. Remember that other mitochondrial protein, OPA1, the one involved in mitochondrial fusion?
Yeah, OPA1. It's a big player in keeping those mitochondria in tip-top shape.
Right, and it seems like FM162A might be playing a role in regulating its activity. And you touched on it earlier, the importance of that delicate balance between fusion and fission for mitochondrial health.
Oh, absolutely. Mitochondrial dynamics are critical. Think of it like a constant dance. a merging and dividing of these organelles, essential for their function. Fusion helps repair damaged mitochondria and share resources, while fission gets rid of those that are beyond repair.
So it's like a constant recycling and renewal process going on inside our cells. And when that balance gets disrupted, that's when we start seeing problems.
Precisely. When that balance is off, we see a buildup of dysfunctional mitochondria, and that's a hallmark of aging and age-related diseases. So anything that can help maintain that balance, like this FM162A, well, that's potentially a big deal.
And this paper didn't just look at the mitochondria in isolation. They also found a connection to the mTOR pathway, which, is another big player in the aging game.
Ah, yes, the mTOR pathway, that intricate signaling network that influences cell growth, metabolism, and yes, aging. So what's the connection with FAM162A?
Well, they found that FAM162A seems to inhibit the mTOR pathway. And we know that overactive mTOR can contribute to accelerated aging.
Makes sense then why the flies with increased FAM162A lived longer and healthier. It's like putting a check on that overactive mTOR.
Yeah, pretty fascinating stuff. It really shows how these seemingly separate aspects of cellular biology are all interconnected, right?
Absolutely. It's a complex web, and this research is helping to untangle some of those threads. But let's be realistic here. There's a long road ahead before we can say for sure whether targeting FAM162A in humans would have the same beneficial effects.
Right, we're talking about a study in fruit flies. Translating those findings to humans, well, that's always the challenge.
Exactly. But it's a starting point, a clue that could lead us down some interesting paths. It definitely warrants further investigation in more complex organisms. All right, enough about flies for now. Let's move on to that second paper, the one that tackled resilience.
Resilience to stressors, a dynamical systems approach. It's a mouthful, but it's tackling a concept that's really important, especially in the context of aging.
Yeah, resilience is something we talk about a lot, but actually measuring it objectively, that's a whole other ballgame.
And this paper takes a pretty unique approach. Instead of relying on self-reports, subjective questionnaires and all that, they went for physiological measurements.
Ah, a more objective approach. I like it. So what kind of stressors did they look at, and what were these physiological measurements?
They didn't mess around. They focused on older adults facing some pretty major life challenges, knee replacement surgery, starting dialysis, even bone marrow transplants.
Wow, those are some serious stressors, not your everyday kind of stress.
Right. Talk about a resilience test. They measured things like heart rate variability, which tells you about the balance between the sympathetic and parasympathetic nervous systems.
So how the body is responding to stress on a very basic level.
Exactly. They also looked at cortisol levels, a key stress hormone, inflammatory markers, even sleep quality and cognitive function. All those pieces together painted a picture of how their bodies were actually coping with these challenges.
So instead of just asking someone, how resilient are you, they actually looked under the hood to see how their physiology was responding.
Right. And that's what they call the study of physical resilience in aging, or SPRING for short.
Oh, spring. I like it. So what did they find? What did these physiological measurements tell them about resilience?
Well, they were able to create what they call a physiological profile of resilience, basically a snapshot of how an individual's body responds to stress based on those objective measurements.
And the potential applications of that are pretty exciting, right? Think about personalized medicine, tailored interventions, even risk assessment for age-related decline.
Exactly.
Yeah.
Imagine being able to identify individuals who might be more vulnerable to stress, or those who might bounce back more quickly.
Yeah.
That kind of information could be incredibly valuable in a clinical setting.
Absolutely. It could revolutionize how we approach healthcare for older adults. And beyond just clinical applications, think about what this kind of research could tell us about the underlying biology of resilience.
Right. What are the key pathways, the networks, the molecular mechanisms that make some individuals more resilient than others?
If we can crack that code, we might be able to develop new interventions to actually enhance resilience. Not just help people cope with stress, but actually boost their ability to thrive in the face of challenges.
That's a powerful concept. And it brings us to the third paper on our list today, which delves into another hot topic in longevity research, DNA methylation clocks.
Ah, yes, those epigenetic clocks that everyone's talking about. I'm always curious to see what new research is emerging in that area. So what's this paper all about?
It's titled A Comprehensive Assessment of Methylation-Based Age Prediction Methods. And it's a deep dive into the accuracy and reliability of these clocks.
Interesting, because there are a lot of different clocks out there, right? And it's not always clear which ones are the most reliable or appropriate for different research questions.
Right. And this paper tries to provide some clarity. The team from Boston University compared the performance of a whole range of clocks, assessing their strengths and weaknesses.
Okay, so they're doing some head-to-head comparisons. That's always helpful to see. What kind of things were they looking at?
Well, they looked at how well each clock predicted chronological age in different data sets, how consistent the results were across different populations, and they also delved into the limitations of each clock.
Good, because no tool is perfect, right? It's important to understand those limitations.
Absolutely. And they really emphasize that point, that just because a clock performs well in one population doesn't mean it will be equally accurate in another.
It all comes down to how those clocks were trained and validated, right? What kind of data was used, what populations were included.
Exactly. And this paper provides researchers with a sort of roadmap, helping them navigate the methylation clock landscape and choose the right tool for the job. Because it's not a one-size-fits-all situation.
No, it's not. And as these clocks become more widely used, not just in research, but potentially in clinical settings, it's crucial that we have a clear understanding of their strengths and weaknesses.
Right. We need to be able to interpret the data responsibly and make informed decisions based on it. So this paper is a valuable contribution to the field. It's bringing some much-needed clarity to the world of methylation clocks.
I agree. It's a good reminder that while these clocks hold immense promise for longevity research, we need to use them carefully and critically.
I feel like we've covered a lot of ground already. We've got this potential new player in mitochondrial health, FM162A, this objective way to measure resilience, and a critical look at DNA methylation clocks.
Yeah, a lot to digest. And we've only just scratched the surface. There's so much more to explore in each of these papers.
So much good stuff in these papers. But if we had to pick one key takeaway, what would you say is the most important thing for our listeners to remember?
Well, I think the big picture here is the incredible pace of progress in longevity research. These papers are just a snapshot, really, but they highlight the kind of groundbreaking work that's happening. We're really starting to understand aging at a deeper level, and that's paving the way for interventions that could actually extend both lifespan and health span.
It's inspiring, for sure. But let's go back to something you mentioned earlier about resilience. That paper, with its objective measurements, it really got me thinking. If we could reliably measure resilience like that, how could you, as a researcher, actually use that knowledge?
That's a fantastic question. It opens up so many possibilities. Let's say I'm developing a new senolytic drug, right? Instead of just looking at age, I could use resilience profiles to select participants for clinical trials. See if the drug is more effective in people with higher resilience, maybe even tailor the dosage based on their profile.
Interesting. So resilience could be a much better indicator than just chronological age.
Exactly. And it goes beyond clinical trials. Think about preventative medicine. What if we could identify people with lower resilience early on? Maybe we could intervene with lifestyle changes, targeted therapies, give them that extra boost to prevent age-related decline.
It's like personalized medicine, but for resilience. Tailoring interventions to each individual's needs.
Precisely. We're moving away from a reactive approach to healthcare, waiting for things to go wrong. Instead, we're being proactive, focusing on prevention, building resilience to stay healthy longer.
And it all comes back to understanding the biology, right? Those molecular mechanisms that drive resilience.
If we can figure out what makes some individuals more resilient, maybe we can even develop therapies to enhance resilience in everyone. That's a game changer.
It's exciting to think about the possibilities. So as we wrap up this deep dive, what's your final message for our listeners?
Stay curious, stay engaged, stay connected. Longevity research is moving fast, and it's through collaboration, sharing knowledge, that we're going to make real progress toward extending healthy lifespan for everyone.
Well said. And that's a wrap for this week's deep dive into the latest longevity research. We hope you found it insightful, maybe even sparked some new ideas. Until next time, keep exploring, keep pushing the boundaries. We'll be back soon with more fascinating findings from the world of longevity.